Deoxyribonucleic acid, better known as DNA, is a fantastic molecule, and much of modern biology is centered around studying it. All living things are made of cells. DNA acts like an instruction book for cells and is found inside a special part of the cell called the nucleus. DNA contains a code that tells cells how to work, live, and multiply. The structure of DNA makes it easy for the cells to read and use the code. The structure and shape of DNA was discovered through the work of 3 scientists, James Watson, Francis Crick, and Rosalind Franklin. Thanks to their work, we now know that DNA has a double helix shape. This looks a little bit like a spiral staircase. The backbone of DNA is made out of phosphates and sugars, which look like a handrail, while the DNA base pairs look like steps leading up or down. The steps of this DNA staircase are made of 4 DNA bases, A, T, C, and G. In this staircase, there is an important rule about the steps called the base pair rule. A always connects to T, and C always connects to G. This base pairs rule is important because it allows cells to copy DNA easily or repair it if it gets damaged. This is important for when our body needs to make new cells and to keep cells doing their job properly. These bases make up the DNA code. Cells can read these bases like words and sentences, which we call genes. When cells need to make new parts, they copy the instructions from the genes and send them as a message outside of the nucleus. This message is sent to tiny factories within the cell, where it is read and used to build what the cells need. Because DNA acts as a code of instructions, studying DNA helps us to understand how cells work. Scientists who work with DNA are called molecular biologists. Because DNA is very small, 1,000 times thinner than a strand of human hair, scientists who do experiments with it need to work with very small amounts of liquid. They use 2 important pieces of equipment to do this: micropipettes and microcentrifuges. Micropipettes are used to work with very small, precise amounts of liquid, as small as 100 microliters. That is 1/10 of a milliliter. Microcentrifuges are used to separate out small things that are inside cells. These machines spin the contents of the opened cell very fast, and this helps to separate out different things inside the cell, including DNA. Extracting DNA from cells is something that molecular biologists do all around the world, as it is the first step of most experiments. And you can do it too at home or in the classroom. Once the DNA is extracted, scientists can do lots of different things with it, using special laboratory techniques. They can find out the order of the letters. This is called DNA sequencing and helps scientists to read the DNA. They can also cut out specific parts of the DNA that they want to study. They can look at the size of these different parts of DNA by separating them in a gel. They can also make copies of the DNA, called amplification, using different techniques. And then can make changes to the letters, which will then change the instructions. Of course, nothing will happen if the DNA stays in a tube. To study the effect of the changes made to DNA, scientists first have to put it back into a cell. The cells, which can be cells from bacteria or humans, are grown by scientists in the lab. They cannot survive outside the lab. These special tools and techniques help scientists all around the world to do many different things with DNA, either to study it, to learn more information, or to make useful products. For example, scientists can study genetic diseases. These diseases are caused by permanent mistakes appearing in DNA instructions, which can cause the cells not to work properly. An example of this is cystic fibrosis, which is caused by a change in the DNA. This causes a very sticky mucus to build up, which clogs the lungs and makes it difficult to breathe. Scientists can also study the DNA of plants or animals for agriculture. For example, studying the information of cattle DNA can help scientists to figure out which food is the most efficient. This helps farmers to decide on the best diet to give to cattle according to their DNA information. This allows them both to increase profit and reduce the impact on the environment. Scientists can use DNA to help cure people, like people suffering from diabetes, for example. Cells from their pancreas cannot make insulin, which is a vital substance in your body that helps control sugar levels. In order to survive, people with diabetes need to inject insulin into themselves. It used to be very difficult to get this insulin, but it can now be easily made in laboratories by bacteria. All we had to do was to give bacteria the human DNA that contains instructions for insulin so that they can make it. In 2020, scientists were able to quickly develop vaccines to fight the COVID-19 pandemic. The vaccines were made using molecular biology techniques that scientists had been already using for years. This meant that a vaccine using this technique could be developed quite quickly.
Some of the COVID-19 vaccines contain DNA or a message made from DNA, which tells cells in human bodies how to build a small part of the SARS-CoV-2 virus responsible for the disease. Once made by our cells, these small parts of the virus are used to train our immune system at recognizing the virus. Then, if the SARS-CoV-2 virus enters the body later, the immune system will recognize it by the little parts it already knows and destroy it quickly.
These are just a few examples of the amazing things that scientists can do with DNA instructions. We are constantly learning more about how we can apply this knowledge to improve the world we live in. Maybe one day you can make a new DNA discovery.